Good morning everyone. Today we are going to study about the urinary system, the histology of urinary system, how the kidney, ureter and the urinary bladder can be studied under the microscope. What is its normal structure? What are the different features which can be seen under the microscope? This is a study which we are going to learn about today's class. First we will learn about the kidney. So as we all know, kidney, it is the organ which filter the urine or which creates the urine, which filters the blood to make the urine. It is a bean-shaped structure which have got two in number. Two right and left kidneys are there, which are separated, which are present at the posterior wall of the body. The kidney is made up of nephrons. The single unit of kidney is known as nephrons. The nephrons have got elements named as proximal convoluted tubules, distal convoluted tubules, glomerulus and collecting tubules or the collecting ducts, Bowman's capsule, loop of Henle. I think all these words are familiar to you. So we are not going to study about the normal gross anatomy of the kidney. Instead, we are going to study how it appears under microscope. First, the general features about the kidney. The kidney can be divisible. Under the microscope, the kidney can be divisible into two. That is, here you can see there is the outer cortex, outer cortical region and inner medulla. A cortex and a medulla. We have studied in case of lymphatic system when we have discussed. We have studied there is differentiation of the tissue into cortical region and medullary region. Like that, in the organ of the kidney, in a section, the structures which are present towards the external aspects, they are named as the cortical region or the cortex, and which is present towards the inner core, that is the medulla. So outer cortex and the inner medulla. How it appears under the microscopes. In this diagram we can see this is the appearance of a kidney under microscope. Appearance of the kidney under the microscope. Here the section itself shows a number of cut sections of PCT, DCT and collecting ducts. These are the major contents which can be seen in it. Like that, there is also the glomerulus. The tuft of capillaries can be seen inside the glomerulus. That is also can be seen in relation with the cut section of kidney. So, the PCT, the proximal convoluted tubule. The proximal convoluted tubule. See, in this picture, we can see the rounded structures, nearly rounded cut sections are present with a lining of cuboidal epithelium and the central cavity inside it. That is, the lumen of these rounded structures are nearly oval or rounded in nature. They are small. So they are known as proximal convoluted tubules. They are lining with either cuboidal or flat cells. Like that, there is another set of cut sections are specially drawn in flattened oval shaped rounds. They are the distal convoluted tubules. The distal convoluted tubules can be seen or identified as elongated cut sections. They are oval in nature. The lumen is also elongated. They are also lined with cuboidal epithelium or low columnar epithelium, cuboidal or low columnar epithelium. So, there are the cut sections of proximal convoluted tubules which are small in shape, and distal convoluted tubules, they are elongated. So, the cortex majorly contains these two components along with the glomerulus. Glomerulus is the cut section of tuft of capillaries with a capsule, that is a Bowman's capsule or outer covering. So, three components can be seen in the predominantly two components, proximal convoluted tubule and the distal convoluted tubule, along with one or two or less number of Bowman's capsule also can be seen on the cortex. In the medulla, there are the collecting tubules, collecting ducts or the collecting tubules are there. The cut sections of collecting tubules can be seen in elongated nature, elongated sinusoids like elongated a large gap areas can be seen. This is specially arranged which are projecting towards the cortex. They are specially arranged elongated gaps. They are also lined with flattened cells with nuclei, and this space are different in nature, that is collecting tubules at different sections can be seen in the medulla. They are named as medullary rays. These medullary rays are the collection of cut sections of collecting tubules which are projecting from the medulla to the cortex. They are named as the medullary rays. So, the major four components or features can be seen in a cut section of kidney are distal convoluted tubule, elongated lumen with a oval shaped lumen with lining, epithelium, and proximal convoluted tubule. They are small rounded with rounded lumen and Bowman's capsule with a small space, like a small vacant space, and there is the collecting tubules. They are elongated spaces. So, now we will learn what are the notes we have written in the slides. The kidney can be divisible into outer cortex and the inner medulla. That I have told you, it can be divided into outer or divisible into outer cortical region and inner medullary region. The cortex shows clusters of renal corpuscles, cut sections of renal tubules like PCT and DCT. Same thing I have told you. That is clusters of renal corpuscles, the Bowman's capsules, cut sections of renal tubules also can be seen. Presence of medullary ray which extend to cortex formed by the aggregation of collecting tubules and the ducts. Collecting tubules and the ducts, they are forming as the aggregations of collecting tubules and ducts can be seen in the medulla. The medulla containing collecting ducts of various sizes with renal tubules and capillary networks. The specialized capillary networks which can be seen in the kidney is known as the vasa recta. It is a network of capillaries can be seen in the kidney, that is vasa recta. That is also can be seen in the medulla. It's not that much visible, but the blood cut sections of blood vessels also can be seen in the medulla. Then how it appears under the microscope, that is cut sections of PCT and DCT are present in the cortex. PCT is larger, oval, irregular lumen lined with cuboid or low columnar epithelium. Specialty of the epithelium, it is brush bordered. We have drawn the brush bordered epithelium, that is a microvilli. Brush bordered. The cells are large. DCT is small rounded lumen cuboidal epithelium. That is, they are small in nature and they have got rounded lumen and lined with cuboidal epithelium. The cut sections of loop of Fenley can be seen as oval spaces lined with flattened epithelium. That is, cut sections of loop of Fenley can be seen like oval spaces, simply oval spaces. Medulla contains collecting ducts with a round lumen and flattened cuboidal epithelium lining. Medulla contains collecting ducts are there with round lumen and flattened cuboidal epithelium lining. So that is the collecting ducts or the collecting tubules, cut sections of collecting tubules which are forming as a medullary ray. So this is how it appears under the microscope, and this is the diagram how it shows under the this was the schematic representation of the kidney under microscope, and this is how it appears under the microscope. Here you can see the small rounded structures. Rounded, they are the PCTs, and elongated spaces, they are the DCTs, the distal convoluted tubules, and there are blood vessels also can be seen, and this rounded spaces represents the glomerulus, that is the renal corpuscles. Then medullary ray, you can see the medulla is extending as elongated spaces into this, that is the medullary ray. Into the cortex it is extending. So this is how it appears under the microscope. Next is the ureter. The ureter is a tubular structure. There are right and left kidney like that. From the kidney, this tubular structure which connects to the urinary bladder is known as the ureter. So the ureter, right and left ureters are there. It is about 25 centimeter in length. So they are thick-walled tubular structures. Now we will learn about the histology in the section. This is how it appears under the microscope. It is a tubular muscular tube. It's formed of a muscle. The smooth muscle is the muscle forming within the ureter. That is, we have studied three types of muscles: the striated muscle, smooth muscle, and the cardiac muscle. So out of which striated muscle or the skeletal muscle, they can be seen in relation with the bones. The smooth muscles are present. The smooth muscles are present within the walls of the blood vessels, within the walls of the organs which have got cavities in it. So here is also there is a presence of a cavity or lumen can be seen inside the ureter, so that for the passage of the urine, isn't it? It is a tubular structure. So for the passage of urine, inside that there is a lumen. So this is also lined with the wall of the ureter. It's also lined with smooth muscle, and the ureter have got inner mucosal lining. That inner mucosa contains two structures. Mucosa means that is the two structures which are forming with the lining epithelium. We have studied the lining epithelium of urinary structures as urothelium or transitional epithelium. When we have studied about the epithelium, we have learned transitional epithelium. So the transitional epithelium is the structure which lines or the epithelium, specially forming 5 to 6 layers. By innermost layer is the cuboidal or log columnar. Above that, pear-shaped or the polygonal cells, 2 to 4 layers are there. And outermost there is the presence of umbrella shaped cells. So that is, this type of epithelium can be seen in this near towards the lumen.